a company Pharma and from Aromics, which is another small CRO from Barcelona here. And we have done joint research in the recent few years. And just recently we spun out a company called Aces Therapeutics, which had the task to find money to develop our final product. The time. Okay. So a bit of background history, first of all. So timidate synthase is a key enzyme for cancer treatment, and is a key enzyme which is involved in the DNA synthesis and cell proliferation and represents one of the best known drug targets in the anti-cancer area. Years ago we were searching for money, of course, and we joined three consortium submitting research project to European Commission. Generally you have 50% chance to be successful. So two projects were discarded and one was accepted. The one was accepted was in timidate synthase, so we start to do some research on timidate synthase because we had never done before. And this is quite peculiar enzyme because the enzyme is in the monomer form is not active. It dimerizes and create a catalytic site and become active. Then, which is peculiar, is that the monomer is in equilibrium with this mRNA, and so the activity in the cellular levels of the compounds of the enzyme depends of the binding of the equilibrium with this mRNA. When you go with some enzyme inhibitors, the cells feel that there is no more activity, so the monomer detach from the mRNA, dimerize, and the mRNA produce more protein. And the result that you increase resistance in this sort of tumors, and the expression of TS predicts the outcomes of patients. And we see here that two types of cells treated with famous TS inhibitor, 5-FU, and if after the treatment the level of TS increased in the resistant cells. So there is an obvious need for new compounds able to interrupt this antitumour production in cancer cells. Chu, which is from the States, a famous expert in timidate synthesis, showed that antisense oligonucleotide or small interfering RNA are able to restore sensitivity to cells with high level of TS, most probably by binding to the mRNA and prevent the synthesis of new protein. So a new innovative mechanism for caption in place of enzyme inhibition could be targeting translational autoregulation of timidate synthase in mRNA. And then we became aware of this paper by Rando that use minor group binders which are able to bind the mRNA, and he reported the study, binding studies, and we were quite interested in that one because distamycin has been an antiviral agent discovered in Italy, from Italian, and they work a lot on distamycin, so I was quite interested in that one. We tried this compound on cells, and we found regretfully that all these compounds are more active on sensible cells rather than on resistant cells, so are quite useless from the therapeutic point of view. And this is we saw that the level of the enzyme and the activity of the enzyme, they do correlate. Then we go back to berberine, and we tested the effect of berberine on TS expression, and we found that contrary to the previous compound, berberine was more active cytotoxic of resistant cells than on sensible cells, and this correlates with the activity, with the level and the activity of TS, you can show here, and you see that by increasing concentration of berberine, we especially in, above all in the resistant cells, we have the level of the protein, the reduction is decreasing, and parallelly also the activity. So what is berberine? Berberine is a beta-tyrosine isoquinolina quaternary alkaloid, which is extract from plants from several genera. This is the synthesis. These are the yellow flower, which characterize the plants berberis. And this is a footpath. I call the berberine footpath. This is nearby my house in Italy, North Italy. And all these plants are of berberines. And these are the the red berries, because the flowers turn in red berries at the end of the season. This is my wife with our dogs. So I just discovered recently that behind my house there is this path full of berberine plants. And it's used since 100 years in the Ayurvedic, Chinese, and Native American medicines. And this is a pleiotropic substance, which has diverse pharmacological properties and activities. It's antimicrobial, antidiarrheal, anti-inflammatory, antiarrhythmic. Is a good agent to lower cholesterol, and also is also anti-tumor. Why it's active, we really don't know. It's still a matter of debate. We think that modulation of protein expression by interacting with nucleic acid is a main mechanism of action of this compound. However, the interaction between berberine and nucleic acid has been reported since 1962. And if the alkaloid is a DNA minor group binder, as reported by Rosanna Mondelli after an MR study here, or it is an intercalated DNA, is still a matter of debate. So this alkaloid has a definite potential, in a wide spectrum of clinical application, including hyperlipidemia, metabolic syndrome, polycystic ovarian syndrome, obesity, fatty liver diseases, coronary artery diseases. However, the compound has drawn most extensive attention in these therapeutic areas, and at the moment there is about 23 on clinical studies, and in a few years ago there were only 9 studies. And we published a review about this new perspective for all remedies if someone is interested to read it. Anti-cancer properties, however, berberine in preclinical studies has been widely published since many years. We review this activity of berberine, and Feng published just a few days ago another paper about that one. However, there is no clinical trials in this area, in the anti-cancer area presently. So our finding is this one. Berberine for us represents an interesting and attractive natural lead compound. We can introduce chemical modification in order to have a compound with better effects or different effects. And we perform a new chemical modification on the berberine structure, and we obtain a new class of compound derivatives which are characterized by having linkers of different functionality and length, bound to specific positions on the berberine, and this bring aromatic moieties of different specificity, because in order to create a geometric propensity for additional stacking type non-covalent aromatic interaction, because in biology aromatic interaction are very, in nature, are very important for recognitions. This is our chemistry. We use known chemistry, which are this start from berberine. Berberine is transformed in a sort of derivative of the berberine, and this is alkylated with the moiety we want to insert in the 13th position. Alternatively, we discovered this old and common aldehyde in amine condensation. So we start from the hydroberberine, and I think the better is going on. We go from. We got another one, sorry, because the battery. Okay. So we can functionalize the position trend with this reaction with generally good yields. We tested this compound to see if we improve the recognition and the binding with the DNA. And in this class here, we see that the binding, they do bind to the calf-thymus DNA much stronger than berberine. They reach a maximum here and then they go to plateau. Whereas in this series here, the diphenyl series, still they do bind to DNA better than berberine, and they have just a different profile. Whereas with the pyridine moiety, they do bind better than berberine, and they follow a growing profile like that. Then we went to we turned our attention to mesothelioma because this is a fatal asbestos exposure associated cancer, and it affects the pleural cavity, peritoneal area. The survival time is very, very short, and there is a red tumor, but with an increasing incidence worldwide, and it's supposed to reach the peak in 10 or 15 years. And this is characterized by high levels of T mediator synthase protein expression. And patients which have this marker are non-responsive to pemetrexed and cisplatin. And we found a paper by Koropati very interesting, saying that talking about therapeutic potential of antisense oligonucleotides to downregulate T mediator synthase in mesothelioma. So we test our compound on 3 type of cells, 1 pleural and 2 abdominal, peritoneal mesothelioma, and we see that our compound were much more active than berberine and showed a very interesting activity on this sort of cells. Then we measured the level and the activity of T mediator synthase with respect to berberine, and we found that in these pleural mesothelioma cells, some of our compounds inhibit the synthesis of T mediator synthase to a greater extent. After a series of studies, we choose this compound here to go further. It's NAX35, and these are some data about the antiproliferative activity on chemo-resistant humans mesothelioma cell lines in comparison with standard, and we see that this compound has very good and very interesting activities, and moreover this overcome pemetrexed and cisplatin resistance in mesothelioma cells. The compound was tested in vivo on this STO peritoneal mesothelioma. STO cells are taken by a patient. This experiment was done by Fondazione IRCS, Istituto Nazionale dei Tumori in Milano, famous tumor institute, and the compound was tested IP and PEROS. This is very important because it was very active by PEROS at 10 milligram and 15 milligram. 10 milligram kilo, and much interested that we correlate in vitro and in vivo TS protein levels. This is an in vitro study. We see that the compound inhibits synthesis of proteins in vitro, and in the same cells in vivo, the compound moderates the synthesis of T mediator synthase in treated animal compared to untreated animals. So, concluding with this first product, NAX35 is an innovative proprietary compound which is structurally related to the plant isoquinoline alkaloid berberine, as a novel mechanism of action which targets the expression of TS proteins, different from previous TS inhibitor. As an efficacy of chemo-resistant tumors, and this efficacy is at tolerated doses and is by oral and IP administration in human mesothelioma xenografted model. Now go back to breast cancer. Breast cancer, HER2 positive breast cancer represent 20-30% of invasive breast cancer associated with more aggressive disease progression and poor prognosis. These are the compounds which are presently in clinical use, and these drugs seem they show modest efficacy. So we need new agent which exhibit a new mechanism of action different from the mechanism exhibited by current drugs. We found that berberine inhibits cellular growth of breast cancer cells and promotes apoptosis, but down-regulates these pathways. And there's the two papers which found it. So we tested some of our compound on the cells, murine and human cells, which express HER2 receptor. And we found that these four compounds, particularly interesting was NAX14, which has a very good activity, whereas berberine was almost inactive. 35, which was the compound very active on mesothelioma, was not active, and NAX30. However, NAX13 and NAX14, you see the difference is just that one has a chlorine disposition, the other one is a retox, but the activity is completely different. One is active, the other one is not active at all. So small chemical modification may affect really the biological behavior of a chemical compound. This is the time-dependent antiproliferative activities of this compound here. And in the hand, we choose the NAX14 as the best lead candidate. And we see that this compound here, NAX14, sorry, come here, inhibit the synthesis of H2, with an extra, for instance, to trastuzumab, which is complex the protein but not inhibit the synthesis, whereas berberine and NAX12 they do not inhibit the synthesis. And this was published there. And this again, I would like to point out that small chemical differences may affect the biological behavior. And this compound here, NAX14, was tested on this breast cancer, H2 positive breast cancer model, which is very interesting because these are transgenic mice which spontaneously develop tumors in the time. And we see from here that NAX14 is by IP administration is active in the decrease in the number of tumor, in decrease in the inhibiting the growth of the tumor, and in the decrease the number, it increase the number of mice free of tumor. And these are not days, but these are weeks, so it's about 6 months time. And NAX14 result is less toxic than berberine itself, as you shown here. And NAX14 is also affected by oral route, as was the NAX35 in the previous test, and show an interesting anti-metastatic effect, much with respect to control. And by oral route, inhibit the growth of tumors and even the number of tumor per mouse. And this showed to have also some control of the vascularity. And we show here these are untreated animals. This is animal treated with berberine as a control, and these are animals treated with NAX14. So as some sort of anti-angiogenic activity as well. So many combined mechanisms which can be useful and positive. So NAX14 is again an innovative proprietary compound, structure related to isotretinoin as an ability to reduce H2 expression within via this postulated novel mechanism, whereas current drugs either act on the receptor or they do act kinase pathway, we think that NAX14 acts on the expression of HR2 from nucleic acid somewhere. It's a compound which has anti-cancer, anti-metastatic efficacy, and there's a good tolerability in vivo, even oral administration. So, conclusion, berberine exhibits diverse pharmacological properties in a wide spectrum of clinical application. That might prevent the use of this compound, this natural compound, as a drug for a definite therapy, because there's so many activities, and one say, okay. However, for a chemist, the structure of berberine represents a very interesting biological skeleton, very interesting from the biological point of view, and as an attractive natural compound for a chemist anyway. And if one performs rational chemical manipulation, they can obtain products which are selective for particular therapeutic areas. And by doing chemical modification at operating position, one can discriminate the activity and the medical indication. So we think that berberine, in a specific way, suppress natural protein synthesis, and by appreciation of differences in mRNA translational content between normal and cancer cells. This data are from us, and we see that mRNA, mRNA, in presence of berberine in sensible cells and in resistant cells do behave differently with respect to control. And we see that in this case, mRNA in presence of berberine in resistant cells is more stable than in control. It means that some sort of complex should be happening between the compound and the mRNA. This was reported by us a couple of years ago, and also last year was reported by this Chinese group at the recent Congress. However, we showed before that NAX14, but not NAX12, do inhibit synthesis of HR2 protein. And in that case, we saw that NAX45 prevent the synthesis of dimethylate synthase, but NAX14 doesn't prevent the synthesis of dimethylate synthase, but it does prevent the synthesis of HR2. So by playing with some chemical modification, I think this compound will be very useful in future to block the synthesis of some specific proteins which are relevant from disease point of view, tumor or other diseases. Future perspective, we investigate also the effect of the berberine derivative of colon cancer cells. We just published twice, and we found that berberine and our derivatives are an inhibitor of the signaling pathway between Wnt and beta-catenin. So they do repress the Wnt expression. And we found some activity also in hepatocellular carcinomas, cell lines. This is very interesting because berberine is used clinically in China for to cure as a natural integrator, as a natural product, hepatocellular carcinoma, which is very widespread in China. And we found also that this compound recognizes human telomeric G quadruplex. This is a paper we published, and this is one we are going to submit. We obtain X-ray complex, the X-ray resolution of the complex between one of our compound and these telomeric sequences. And this is an area we have to investigate to see what happened. Now I'm finished. Now I would like to make acknowledgements and financial support were provided by Italian Ministry of Economic Development with this grant, and from the Agenzia per la Competitività dell'Impresa, Axio here, which is which is a department of the Catalonia Municipalidad de Catalunya for financing the companies in Catalonia. And this was done under the 6th call for the Eurotransfer initiative, which is a transnational project berberine as antitumour agents. Okay. Thank you.
Thank you.
Thank you. With our compound, you say? Toxic effect and berberine has been used, as I told you, since many hundreds of years. And you can go on internet and buy berberine. And for the personal consumption, there is the American, I think they use a lot because most of the sites come from the United States. And the most important thing is that when you do a chemical modification of the natural compound, you improve the toxicity or reverse the toxicity. In that case, I show you that with our compound, especially the compound is active on breast cancer, we got a compound which is less toxic than berberine, and this is good. We would like to go into the clinic with these two compounds, NAX 35 and NAX 14 are our clinical candidates. And we have a business plan to go there. We are looking for funds, which is not easy to find, especially in drug discovery. It's not easy to find, maybe it's easier to find in the States, but it's not easy to find in Italy for that one. But we believe very much in those two compounds here, and we would like to go further. And we will be the first-in-class compound, this one, because there is no berberine in the clinic used as anti-cancer.
Actually, I have a couple of questions. So, number one, you showed the very impressive picture how your compounds decreases the density of blood vessels in the human.
Yes.
So, do you have any idea about the mechanism of this anti-angiogenic effect?
No, I don't have an idea because we don't have biology in house. We have to resort by external collaboration. I think they are studying and probably they will produce a record of that. These are data they show us, and which are data that add to the antitumour activity of the good compound. Even doxorubicin has some sort of anti-angiogenic activity. I mean, I think that the natural product has one principal activity and then a lot of collateral activity.
Okay. And another more practical question. What dosage form and what route of administration do you envision for this one?
You mean in first-time men?
Yes.
We have to do all the non-GMP and GMP standard increasing toxicity before to do that one. But I think that I can order by oral route or by IP. It depends on the tumor. Peritoneal mesothelioma is treated locally at the moment, so you can also think about local injection. We really don't know. We only know that the compounds are orally active, which in some tumors, to treat some tumor may be beneficial, that one. For others, it's completely.
Yes, please. Yes, Ben.
I saw there was a publication relating to P53, and some mesotheliomas have P53 mutations or overexpressions.
Sorry, is that publication related to?
P53, P53
Yes.
overexpression, and some of the mesotheliomas also have P53 mutation or overexpression. And it will be interesting to see if you have any data in the future for ovarian cancer, because of high risk, high grade ovarian cancers are p53 driven, and if the IP injection could be useful, that would be useful for ovarian
Yes.
cancer too.
Yes, we have some data. As a matter of fact, when we test berberine on those first cells, they were ovarian cells. I put between quotes because apparently it's not like that. We had some tests, in vitro with yes, some resistance ovarian cancer cells, and the product are active. Yes, this is an area to investigate.
So the last question.
Yes,
Yeah.
Thank you, Dr.
Right.
Yeah, I think if you make comparison between the pharmacokinetics after oral administration, this will be very strong point in the future, because one of the major limits in cancer therapy is the route of administration.
Yes.
This point number one. Number two, about the answer, it can reduce the angiogenesis. You mentioned that it inhibits the Wnt-catenin pathway. Okay.
Yeah.
So I think there is direct correlation with many features of cancer, including the angiogenesis. So maybe further by measurement of the vascular endothelial factor or something like.
Okay. Thank you. Yes.
Okay, so I think we are done with